Hello everyone, it's glad to be here. And then today I will be presenting something a little bit different. It's called sodium zirconium phosphate as the absorbent material for ammonia removal in animal cell culture system, which can be potentially used in a media recycle system. Okay. For those you have not been to University of Alberta, I am an assistant professor at University of Alberta, very new. And University of Alberta is located in Edmonton, Canada. And then the campus is really beautiful and large. And Edmonton is famous for its long winters. But from a bright side, it provides a very excellent environment to focus on research. Okay. Yeah. So let us go to the theme. One of the major challenges today for cultivating meat production is the high cost of media, cell culture media. So up to 50% of media is in the spent cell culture media is remain unutilized. And then cell culture media is always expensive. Therefore, doing recycled media technology can provide a variable solution to reduce the cost of the media. And however, in order to do the recycled media, we always need to solve some challenges. One of the main challenges for doing a recycled media is to solve the problem of accumulation of toxic metabolic byproducts from the media. Ammonia is one of the main challenges in animal cell culture media system, especially in high cell density and prolonged cultures. In cell culture media, the media is always from the range from 7 to 7.4, where the ammonia is in its ammonium ionized form. And ammonia is a toxic bioproduct from animal cell culture bioproduct. And then it can inhibit the cell growth, variability, and then productivity at really low concentration, as low as 2 millimole. And for this purpose, removal ammonia in the spent media is very important for doing recycling media. Current strategy, there are a lot of strategies for ammonia management in biopharmaceutical industry. For example, there are media optimizations, establish perfusion system, develop cell lines which can have more effective usage of the nutrients or reduce the metabolic pathway of ammonia production, and also establish pH control system. And also, by all this method, they have some limitations. For example, they have very complex operation system, or they are expensive, and also some of them have regulatory considerations. So they have some limitations on cultivating meat production. However, there are also some methods currently are used. There are research on adsorption-based removal. This method, they have some limitations on capacity and selectivity. However, they are simple to operate, and then they are cost effective. And here are some methods already being used in cell culture or cultivating meat production. For example, ion exchange resin has been used for removal ammonia, and also use of the active carbon. This method, they are either use ionic exchange method or they are using some absorption method. They have low selectivity for ammonia. And then there are also some method using zeolites artificial zeolites. For example, one patent from Future Meat Technology or Believer Food, they have one patent on using this method to incorporate with their hollow fiber system to establish a media recycle system, reduce the cost. Therefore, this method has potential in using in media recycle system in cell culture meat production. And however, there are also some researches on zeolite research showing that there could be slight cytotoxicity of artificial zeolites. And here we introduce a material called zirconium-based material. Zirconium hydrogen phosphate, they have already been used in ammonia adsorption for some researches. They have very interesting structures. They have an interlayer spacing of 7.6 angstrom, and in between they have some hydroxyl groups which can induce ion exchanges. So 7.6 angstrom can eliminate some large molecular going inside their interlayer to, for example, some amino acid, they always have a larger size, more than 1 nanometers or 10 angstrom. They cannot go inside into the interlayer spacing, so they will not do the ion exchange. Therefore, the selectivity of ammonia is high using this method. However, the natural-based zirconium hydrogen phosphate, they are acidic in an aqueous environment. Therefore, they need to be modified in order to adapt to animal cell culture system. Here we did 3 methods with 4 different chemical compositions, and then we produced 4 different types of sodium zirconium phosphate components. And then the results shows that with the first two methods, which is produced by minimal solvent method, the sodium zirconium phosphate A and B, they showed really high ammonia absorption activity. And then for C and D, either they show low absorption or they have changed the pH very much, so they are not suitable for future further cell culture system. And for the reason behind it is that interlayer spacing is very important. So we did an XRD analysis, and we showed that interlayer space between A and B is around 8.5 angstrom, which can allow the ammonia to go inside the interspace. And then for the sodium zirconium C, they show the amorphous structure, which is a disordered structure, and they shows very little absorption activity. And then for the last one, the interspace is smaller than the other A and B, and then they shows less adsorption activity. Then we also did a XRF analysis, and then we showed that A and B has a chemical formula as shown here, and then the 2 hydrogen molecule atom was replaced by the sodium atom, and then the synthetic pathway can also be was also shown here. And for so since A and B, they are very similar, I used sodium zirconium A as an example to demonstrate all the mechanism and behaviors. And here we can see that under Isccm, we can see a very a crystallized layered structure form for zirconium A, and under the TEM, we can even see the layered lattice for this chemical. And then within this lattice, there are these are the place where the ionic exchange reaction occurs. And the ammonia efficiency and rate for the sodium zirconium PA is like we used chicken fibroblast spent media. We collect those spent media, and then those spent media was collected with like 16 passages of the chicken fibroblast, and then the ammonia concentration has already been reached to around 9 millimole, and we used a different amount of sodium zirconium phosphate to test their adsorption efficiency, and then we can see that with the 0.5 gram sodium zirconium, it can absorb more than 50% of ammonia with very little pH change. And then also the adsorption rate is very at 5 minutes it's already reached to more than 50 or 40%, and then it reached to a plateau around 30 minutes, and then it shows a really high adsorption. And for cell compatibility, we're also able to say, demonstrate that this kind of material has really good cell compatibility. It has greater than 90% cell viability after exposed 24 hours with the different concentration of the zirconium-based material. And then the cells also there is very little dead cells, and it showed really good cell compatibility. We also tested the recovery and reusability for these components. So for reusability, we use the ionized water to wash this kind of material after they absorbed the ammonia. And then we see that even after 4 times of usage, they still shows really high non-significant change on ammonia adsorption rate. Therefore, it shows that for this kind of material, at least 4 times, 4 cycles, we can see not too many changes or loss of functionality. And then for the adsorption kinetics, we always can see it here is like within 20 minutes, the adsorption rate is already reached to a plateau, and then it's adsorbed ammonia really fast. And this kind of kinetics also shows that chemical adsorption is the primary mechanism for this adsorption. And for fitting the models, we show that the mechanism is that with a homogenized surface and finite binding site is the adsorption mechanism. And post-adsorption, we always we say that there is a decrease in sodium, which indicates there is an exchange from the sodium to ammonia. And then with adsorption, there is an increase in interlayer spacing. We also proposed a production flow with this kind of adsorption material. So within the production flow of production cultivated meat, and there can be an addition of this fixed bed adsorption column with this kind of material, so that with the recycle system, there can be a removal of ammonia, and then the medium can go back into the recycle system again to reduce the loss of nutrients. And the research is still ongoing. For next step, there are still a lot of things to do. For example, to verify the selectivity and to do a techno-economic analysis. And then this kind of components, the raw material for bulk price is only 0.42 dollars per kilogram, so they're inexpensive. And then it will be interesting to do a media recycle system to see the techno-economic analysis impact. And also to build a continuous flow system will be the next step. For example, to establish a fixed bed system and test the adsorption in dynamic system. And then to do a long-term durability and also broader applications with different cell types and different media compositions. So the conclusion for this research is that we developed a layered based adsorption material which can be used to absorb ammonia in the spent media. And then the absorption rate is as high as 58%, and it shows good cell compatibility and then can be reused for at least 4 times. And then the mechanism for adsorption is selective ionic exchange mechanism. And for this project, we did a collaboration project with Zhejiang University, and then this one is supported by the Good Food Institute. And this is not the last slide. So forgive me, I couldn't find a better place than this to have a recruitment. So I hope, so you don't mind, I take a very short recruitment announcement. There are a lot of things ongoing in Canada. Cultivating meat or cellular agriculture research is thriving in Canada at this moment. And I am currently recruiting 3 PhD students in cultivating meat research. And so if you are interested or if you have students who are interested, please feel free to contact me. This is my contact information. Okay. Thank you very much, and I'm happy to take any questions.
Time for 1 or 2 short questions.
Hi, really nice talk and very useful technology of the moment. So I have 2 questions. Can it absorb ammonia in a smaller quantity, like 1 or 2 millimolar? And the other question is, how long does it take to exfoliate ammonia from the material?
So the first question, can it absorb in a small quantity? Yes. So all the research work we did is in a lab scale experiment. We're collecting those ammonia from flax, so it can be done in a small quantity. And excuse me, what is the second question?
How long does it take to exfoliate ammonia from the material?
It doesn't. So we use deionized water to just wash the material, and then we didn't. So wash the material take like 10 minutes to 20 minutes, and then we didn't say the minimum time to wash it, but just use a lot of water to wash it and to collect it again and reuse it.
Okay, thank you very much.
Thank you.
I think a final question over here.
Very interesting talk, thanks. I had a question around if you've tested like very high concentrations, or particularly at which concentration of ammonium would the material get saturated. And then you mentioned that the viability stays above 90% after 24 hours. How much was the longest time that you tested this for?
Okay, thank you for the question. So the first question is, will this be saturated for the material for high concentration? We haven't reached more than 9 millimolar of the ammonium concentration. I think it's good to learn what is the saturation rate. I think it's a good point to continue the research. And then the second question is,
Said longer than 24 hours.
So we did test 48 hours, but that we use C2C12 cells instead of the chicken fibroblast. It always choose good cell compatibility, like 90%. And then the longest we have done is 48%. And then we also indicate in the future steps it will be good to have a long durability of this test.
Thank you.
Thank you.
Thank you very much again.